Hello friends, I am Dr. U S Desai, working as an assistant professor at PS Modern College of Pharmacy, Nigdi, Pune. So today we will see the gram staining technique, which is one of the type of differential staining technique. So the list of materials required are here. First is a clean glass slide, then is your inoculating loop, which is your nichrome loop, Bunsen burner in order to sterilize the inoculating loop. Beadless paper is it is same like that of tissue paper or filter paper, and it is used to remove the excess of water or stain on the glass slide. Microscope, lens paper and lens cleaner. The lens cleaner is used in order to clean your objective as well as eyepiece lens. Immersion oil. In case of immersion oil, you can use either white paraffin liquid or cedar wood oil. Distill water to clean or wash the slides, and 18 to 24-hour cultures of organisms. The reagents required will be primary stain, that is crystal violet, mordant, that is gram iodine solution, decolorizer, or that is ethyl alcohol, and secondary stain, that is safranin. Now, what is the principle involved in gram staining technique? Application of primary stain crystal violet to a heat fixed smear of bacterial culture. So the crystal violet, that is Cv, dissociates in aqueous solution into Cv plus and chloride ions. These two ions then penetrate through the cell wall and cell membrane of both gram positive and gram negative bacteria. The Cb plus ions later interact with negatively charged bacterial components and stain the bacterial cells purple. The next step is addition of gram iodine solution. Here iodine, that is I minus or I3 minus, acts as a mordant and as a trapping agent. A mordant is a substance that increases the affinity of cell wall for a stain by binding to the primary stain, thus forming an insoluble complex which gets trapped in the cell wall. In the gram stain reaction, the crystal violet and iodine form an insoluble complex, that is Ci-, which serves to turn the smear dark purple color. At this stage, all cells will turn purple. The third step is decolorization with 95% ethyl alcohol. Alcohol or acetone dissolves the lipid outer membrane of gram-negative bacteria, thus leaving the peptidoglycan layer exposed and increases the porosity of the cell wall. The CVI complex is then washed away from the thin peptidoglycan layer, leaving gram-negative bacteria colorless. On the other hand, alcohol as a dehydrating agent acts on the cell walls of gram-positive bacteria, which causes the pores of the cell wall to shrink. The CVI complex gets tightly bound into the multi-layered, highly cross-linked gram-positive cell wall, thus staining the cells purple. The decolorization step must be performed carefully, otherwise over-decolorization may occur. This step is critical and must be timed correctly, otherwise the crystal violet stain will be removed from the gram-positive cells. If the decolorizing agent is applied on the cell for too long time, the gram-positive organisms to appear gram-negative. Under decolorization occurs when the alcohol is not left on long enough to wash out the CVI complex from the gram-negative cells, resulting in gram-negative bacteria to appear gram-positive. Now counter stain with the safranin. The decolorized gram-negative cells can be rendered visible with suitable counter stain, which is usually positively charged safranin, which stains them pink. The pink color, which adheres to the gram-positive bacteria, is masked by the purple of the crystal violet. Basic fission is sometimes used instead of safranin in the rare situations. So now we will see the procedure of gram staining technique. So as we have seen in other staining practicals, first you will require a glass slide which is to be cleaned with water and then with any disinfectant like alcohol. Then you will have to label the slides. Labeling of slides in gram staining is very important so that you can determine whether on which slide you have inoculated gram positive bacteria and on which slide you have inoculated gram negative bacteria. After you label the slides, then you will have to prepare the smear as we had seen in monochrome staining technique, where you first sterilize the inoculating loop or nichrome wire loop by holding it in red flame on Bunsen burner. Once it is red hot, it indicates that the nichrome wire loop is completely sterilized. Then you have to put that nichrome wire loop in a test tube containing your inoculum, and the loop full of inoculum is to be spread on the marked area on the glass slide. Allow it to dry, and after that you have to heat fix that smear, and then you will have to follow the gram stain procedure. So we'll be seeing what is the gram stain procedure. First we will place the slide which is containing the heat fixed smear on a staining tray. Then you will gently flood the smear with crystal violet and let it stand for around 1 minute. Tilt the slide slightly and gently rinse them with tap water or distilled water using a wash bottle. Then gently flood the smear with gram iodine solution and let it stand for 1 minute. Now this gram iodine solution, it is made by dissolving iodine in water. But as you all know, iodine is insoluble in water, so we have to add potassium iodide. So by common ion effect, it allows the iodine to get dissolved in water. So basically gram iodine solution is it is a mixture of iodine and potassium iodide in water. Then tilt the slide slightly and gently rinse with tap water or distilled water using a wash bottle. The smear will appear as a purple circle on the slide. Then decolorize using 95% ethanol or acetone. Tilt the slide slightly and apply the alcohol drop by drop for 5 to 10 seconds until the alcohol runs almost clear. Be careful not to over decolorize. Immediately rinse with water and then gently flood with safranin to counter stain and let it stand for 45 seconds. Tilt the slide slightly and gently rinse with tap water or distilled water using a wash bottle. Blot dry the slide with bibulous paper. View the smear using a light microscope under oil immersion lens. Now we will see the procedural video of how you have to perform the gram staining technique.
See the procedure for gram staining technique. The gram staining technique is usually used to differentiate between gram positive and gram negative bacteria. So the initial procedure of inoculum and heat fixing is same as that we saw for monochrome staining. The area is marked on the slide on which we will be taking the inoculum. This is your nichrome wire loop. You will first sterilize it in flame. You will make it red hot, which indicates that the nichrome wire has been completely sterilized. Put a drop of inoculum in the area marked and allow the smear to spread. Prevent thick smear. If you prepare a thick smear, then the microbes will overlap and clear viewing of the images will not be seen. Now we will heat fix this inoculum by passing this light through the flame 4 to 5 times. Heat fixing helps the microbes to stick to the slide and prevents their washing off or movement. Now you can see that the smear has been completely dried. We will be adding the primary stain, which is crystal violet. We will be adding 2 to 3 drops of this crystal violet in the marked area. And allow it to stain for say 1 minute. So after 1 minute you can see that the stain has completely dried off. We will wash and remove the excess stain. And allow the slide to air dry. Now you can see that the slide has completely dried off. Now we will be adding the gram iodine stain into it. 2 to 3 drops of gram iodine stain. And retain it for 1 minute. Adding the gram iodine stain, we'll wash it off with water and allow it to dry. Now we have completely dried the slide. Now we'll be adding alcohol, which acts as a decolorizing agent. 2 to 3 drops of alcohol on it. Keep it for 30 seconds and then wash it off. Which is safranin, 2 to 3 drops of this. ये तो नहीं sir. You keep it for 1 minute. This is our slide which is ready and completely in dried form. Now you can add cedar wood oil and seal it under 10x objective lens, and you can differentiate between gram positive gram negative bacteria. The gram positive bacteria will appear purple or violet and the gram negative bacteria will occur pink or saffron.
Now here you can see the observation. So the gram positive bacteria will appear purple or violet in color, whereas the gram negative bacteria will appear pink in color. Now here we had used Staphylococcus aureus as an example of gram positive bacteria and E. coli as an example of gram negative bacteria. So in this way you can identify and differentiate between gram positive and gram negative bacteria by using the gram staining technique.